Okay, so what we're getting into now, we have finished Unit 1 with our infectious diseases. Now we're going to get into genetic disorders and genetic testing. That's going to be our focus for the rest of the semester, and it's going to carry into next semester as well. So genetic disorders have to deal with your DNA, okay? Now, the key thing is that our DNA, we have 3 billion base pairs in our DNA. 3 billion. That's a lot. Okay, everybody has 3 billion base pairs in each cell of your body. Okay, think about how much DNA you have in your whole body. That's a lot of DNA. But when we compare us, if we were to look at my DNA versus your DNA, you would see that our DNA is 99.99% the same. Okay, the same, meaning that most of the time we have the exact same DNA. But it's that 0.01% that makes us unique. Okay? Now, it's those changes in our DNA sequence. We're going to look at when something goes wrong with the DNA to cause a genetic disorder. And we're going to look at the different types of genetic disorders in this PowerPoint. Okay, so a genetic disorder is a disease caused by abnormalities in an individual's genetic material. Meaning something is different about the DNA that causes a disease. Now, there's 4 different types of gene disorders. You have single gene, multifactorial, chromosomal, and mitochondrial. Those are the 4 we're going to talk about in here. Okay, so single gene is our first one. Single means one, gene means DNA. So single gene means one piece of your DNA, and it's going to cause a disorder. So it's caused by changes or mutations that occur in the DNA sequence. Now, a gene contains the information for coding for a protein. So remember that central dogma thing that I said wasn't going to go away? We have DNA to RNA to protein. Okay, DNA to RNA to protein. Now, if the DNA has a malfunction in it or has a mutation, that's going to lead to a mutated RNA, which will lead to a mutated protein. Normally, that mutated protein is what causes the disorder in whatever case you're talking about. So, the protein gets messed up and it can't do its normal job. Okay, so with your single gene disorders, these ones are ones that you should recognize from your biology class. So remember, everybody has 2 genes has 2 copies of a gene. You get 1 copy from your mom and 1 copy from your dad. So autosomal dominant, remember we write them like this. Dominant means that it is you have the dominant allele for both of them. That's why we write them as capital letters. Autosomal means it's on a normal chromosome, not a sex chromosome. Then you have autosomal recessive. Autosomal means on a normal chromosome, not a sex chromosome, and it's recessive. So each one contains the recessive allele. And then you have sex-linked. Sex-linked is when it's either on the X or the Y chromosome. Okay, the X chromosome is more common because the Y chromosome basically just says, you're a boy, and that's it. Okay, it doesn't tell you much more than that. It just, that X chromosome is going to be your key there. Now, genetic testing looks at your genotype, and so it will see, are you autosomal dominant? Are you autosomal recessive? Or are you heterozygous? Remember this one? So heterozygous is when one of your alleles is a dominant and the other one is a recessive. And so by looking at the dominant and recessive, that means you're a carrier. Okay, you may not have the recessive disorder, but you do have the ability to pass it on. And hopefully this is all review for you. Okay, so with this one, it says, a man and a woman are both carriers for sickle cell disease, an autosomal recessive trait. What is the risk of having an affected child? Okay, this is a recessive trait, so in order to have sickle cell, you have to be homozygous recessive. Now, the man and the woman, it says, are both carriers. So carrier means you're heterozygous. Now, remember this little tool called the Punnett square? It comes back to haunt you. Okay, so we put our parents on the outside. So we have mom and then we have dad. Then we combine them to see what combinations the kids could possibly get. So this one we could have homozygous dominant, heterozygous or carrier, heterozygous or carrier, and homozygous recessive. So when you look at this to analyze it, we see they have one of the boxes has a homozygous recessive trait. So that means, if you think quarters, 4 quarters equal $1. So this would be a 25% chance. Okay, so this just says 25% chance. Okay, so that was single gene disorders. Basically, there is a mutation in one piece of the gene, so a nucleotide is messed up, and it causes a disorder because it causes a malfunctioned protein. Then you have multifactorial diseases. Multi meaning multiple, factorial meaning factors, multiple factors. Okay, so this is where you have a combination of environmental factors and mutations in several different genes. Okay, so our example here is something like heart disease. For heart disease, your lifestyle plays a big role in whether or not you develop heart disease. Are you a smoker? Do you eat a diet high in saturated fats? All of those could potentially lead to your lifestyle. But we also know that there are some genes that could give you a predisposition, meaning a higher chance of developing heart disease. Then you also have breast cancer. Breast cancer is one where we know that there are certain genes that could lead to breast cancer if they have a mutation on them. But we also know that there's environmental factors as well that play a role in developing any type of cancer. So, multifactorial means that there are multiple factors. There's a genetic component and there is an environmental component in developing them. Then you have a chromosomal disorder. Chromosomal disorder means that there's something wrong with the whole chromosome, okay, which means multiple genes within that chromosome. So humans have 46 chromosomes. You have 44 autosomes. Remember, autosome means that it's any chromosome that is not a sex chromosome. And then you have 2 sex chromosomes. Now, if you are a girl, you have your 2 sex chromosomes are XX. If you are a boy, your chromosomes are XY for your sex chromosomes. Now, the other ones, the 44 autosomes, don't have cool names. They're just labeled 1, 2, 3, 4, 5, etc. Okay, they're not too exciting. But the sex chromosomes, you have XX for a girl and XY for a boy. Now, because chromosomes carry genetic information, if we have extra copies, missing copies, or pieces missing, moved around, etc., that's where it can cause an issue. So to diagnose a chromosomal disorder, we look at what's called a karyotype. Okay, a karyotype, I have a little picture right here. Okay, this is a karyotype. So you have 2 number 1s, 2 number 2s, 2 number 3s, 2 number 4s, etc. Remember you said for each one you get one of those from your mom, one of those came from your dad, because their genes combine together to make up you. Now, if you look on this one, they have 2 X and no Y chromosome. So it is a boy or a girl? Hopefully you said girl. Okay, 2 X chromosomes means it is a girl. Now, we would diagnose this by looking at the karyotype. For example, if you have an extra number 21, so if we throw in an extra 21 there, that's trisomy 21. That's your Down syndrome. Okay, if you have an extra chromosome number 18, so that's trisomy 18, that's Edwards syndrome. Okay, so the extra or missing chromosomes is a whole big thing of DNA. It's not just a small mutation in a gene. It's a whole chromosome that is affected. Then you get into mitochondrial disorders. Mitochondrial disorders are pretty rare, okay? They're not the big ones. So mitochondria, everybody knows you have mitochondria in your cells. They're the ones that look like this. And they're where cellular respiration occurs. Now, these mitochondria make your energy. They're great, but they also have their own DNA. It's called mDNA, mitochondrial DNA. And so with this one, you can have a mutation in your mitochondrial DNA. Okay, that mitochondrial DNA is it will cause an effect, but it's not as common, and it's also not as widespread as a chromosomal DNA mutation. Okay, this is just in your mitochondria. Now, the key thing for mitochondrial DNA is it's only passed from mom to kid, and that's it. Only from mom to kid, because your mitochondrial DNA is the same as your mom's mitochondrial DNA. Okay, so that's your basic for your 4 different types. You have single gene, multifactorial, chromosomal, and mitochondrial, your 4 different types. Now, you can do genetic testing or screening just to see what your genes say, determining if you have a certain disease or not. So, this is the types of genetic testing and screening. The first one we have is carrier screening, and this is one that's going to come up for a lot of you in your scenarios that you're going to get soon. Okay, your carrier screening figures out what your genes say. So it determines whether an individual carries a copy of an altered gene for a particular recessive disease. Okay, so you do not have the disease, but we want to know if you can pass it on to your kids. Okay, so if we know you have a family history of cystic fibrosis, we will probably do a carrier screening and see if you have the gene that you could pass on to your kids. Okay, you can also do it for Tay-Sachs or sickle cell disease. It is looking for that mutation in your genes. Now, you can also do carrier screening. It's the same process to go through if you want to know if you have a certain gene. For example, Huntington's disease, you could look up, you could do your screening and see if you carry the gene for Huntington's disease, because that would tell you whether or not you will develop the disease later on in life. So that is your carrier screening. Now, another thing with carrier screening is that they are looking at your DNA sequence, and they're trying to see what your DNA sequence says. That's the whole purpose of your carrier screening. Okay, now another way to let's say you do the carrier screening and you realize that you have the gene for something. Now, you probably don't want to pass that on to your kids. So you're trying to make a preemptive decision so that your kids don't end up getting sick. So this is where we do pre-implantation genetic diagnosis, or PGD. Okay, now with this is something that is getting into something that is near and dear to my heart, because it pulls in the concept of IVF, or in vitro fertilization. Okay, so with in vitro, I went through in vitro to get pregnant. And so I have a lot of information on how in vitro works because I've been through it. But PGD is not something that we had to do. But we do know couples who have gone through PGD because they don't want to pass on a certain characteristic to their kid. And so PGD, we use in vitro to see if an embryo has a genetic disorder before we implant it back into the mother. Okay, so the way this works, it's easier to show you on a diagram. So here's how this works. You start out in IVF in general is where you take the egg out of the mom, the sperm out of the dad, you combine them together in a petri dish. So that is where you make your embryo. So you put them in a petri dish, you let them grow for 3 or 5 days, and then you implant the embryo back into the mom. Okay, so you take the individual gametes, the egg and the sperm, out of the parents, you put them together in a lab, you let them grow for a couple days, and then you put them back into the parents. Now, that's in vitro. When you throw in PGD, or pre-implantation genetic diagnosis, this is where you have your embryos. Let's take a 4-day embryo. We're going to remove 1 or 2 of the cells. Now, you got to think this is tricky because you only have 6 to 10 cells at this point. So we're going to remove a couple cells. We're going to take out the DNA and we're going to test it for certain chromosomes, make sure it doesn't have an extra chromosome, missing chromosome, where you can do carrier testing on it. We can see its exact genotype. We're going to get the test results back, and if the genetic disorder is detected, we're not going to use that embryo because that's the one that has the genetic disorder. So instead, we put in the one embryo that does not have the disorders, and we put it back into the mom. So you pretty much find out whether or not that embryo has a genetic disorder before you implant it back into the mother to then grow into the baby. So with this one, it's a pretty interesting process. The IVF process itself, just the IVF, is normally between 10 and 14,000 just to do the IVF. But then you throw in the PGD and you're going to spend another 4,000 to 6,000 on top of that because you're trying to figure out if the embryos have the genetic disorders. So a lot of people who have things like hemophilia or cystic fibrosis, or they carry the gene for it, or Huntington's, they go through this process because they don't want to pass that disorder on to their kids. But also, if you have a couple who has gone through miscarriage over and over and they don't know why they keep miscarrying, they will do this and they will find out, is there something wrong with the DNA? Are you having a Down syndrome child over and over and the embryo never grows past a certain time in the prenatal development? And so they'll do this and they'll choose an embryo that is the healthiest and they will implant that one back in because it has a higher rate of working. So that's the process of PGD and IVF. Another thing you can do is once the baby is growing, we have to do some fetal screening. Okay, so this is diagnosing while the baby is still in the womb. So this is where the parents can know what's going on and they can be prepared for it whenever the child comes out. So you have amniocentesis, chorionic villi sampling, or just regular ultrasound are your major ways. And now, in the last couple years, you can do genetic testing. So this is where they just take out the blood they do a blood sample from the mom, and there's fetal cells in it, and they analyze those, and they find out if there's any abnormalities in them. And so all of these allow professionals to diagnose something wrong in the womb before the baby's even born, so that the parents can make the decisions of what to do. Then the last thing, once the child is here, this is where you get into newborn screening. So this is the type of screening where once the kid is born, the state mandates that we screen anywhere from 4 to 30 genetic or metabolic disorder. Okay, we're trying to make sure we catch anything before the baby even leaves the hospital, so if they need to make modifications, they can do so and they can start being prepared instead of finding out later. Because you'll notice in medicine, the faster we diagnose something, we are able to treat it more effectively. So that's your basis of genetic disorders and how we screen for them.